Good morning, everybody. I'm Ryan Farrell, one of the pediatric endocrinologists here at Rainbow Babies and Children's Hospital. But each year, the last day of February is recognized globally as Rare Disease Day, a time to honor patients and families living with rare conditions and to reflect on the scientific progress that makes meaningful treatment possible. In both my personal life and my clinical work, I've seen how dramatically the therapeutic landscape for rare diseases has changed over the past decade. Conditions that once had little more than supportive care now have targeted biologic therapies, gene-based treatments, and realistic hope for altered life trajectories. These advances do not happen by accident. They require scientific innovation, regulatory discipline, and sustained advocacy. We're fortunate today to welcome Dr. Peter Marks, one of the national leaders who has helped shape that progress. Dr. Marks currently serves as Senior Vice President of Molecule Discovery and Head of Infectious Disease at Eli Lilly. But prior to this role, he served from 2016 to 2025 as Director of FDA's Center for Biologics Evaluation and Research, where he oversaw the regulation of vaccines, gene therapies, cell-based therapies, and other biologic products. During his tenure at FDA, Dr. Marks was a consistent advocate for patients with rare diseases, helping to modernize regulatory pathways for gene and cell therapies, and advancing structured communication between FDA, rare disease communities, and therapy developers. His leadership influenced the approval and oversight of numerous transformative therapies, from sickle cell disease and hemophilia to spinal muscular atrophy and Duchenne muscular dystrophy, balancing urgency in the face of unmet need with rigorous standards of safety and efficacy. On both a professional and a personal level, I'm deeply grateful for his contributions to advancing care for children living with rare diseases. Please join me in welcoming Dr. Peter Marks.
Thank you. So thanks so much. Thanks for everyone for coming and for those online for joining. Just by way of full disclosure, my current employment is with Eli Lilly and Company, but anything I say here today are my own opinions. They don't reflect anything from Eli Lilly. Formerly, I was the Director of Center for Biologics Evaluation Research at FDA for about 9 to 10 years. And before that was the Deputy Director for a couple of years. So what I want to do today on this Rare Disease Day week is talk a little bit about the history of rare disease product development, review a little bit of the state of where genetic therapies for rare diseases are. And I'm going to focus on gene therapy, but just understand that a lot of the concepts that I'll present for gene therapies apply to other modalities of therapies, ASOs, siRNA, et cetera. And I'll talk a little bit about what the future state of rare disease therapy may look like and how we might actually try to get there more rapidly, because there are some fundamental disconnects between how we handle large volume products, that is, products that are going to be given to millions of people, with products that are only going to be given to a few people. And I think we have to come to resolution of that disconnect if we're going to make better progress. So just to briefly give you a history of gene therapy, this is like in a thumbnail sketch. The concept of gene therapy was first noted in a Science article by Friedman and Roblin in 1972, this concept that you might use genetic material given to another individual, potentially by a virus. They actually outlined a fair amount of what a gene therapy has come to look like in a 1972 article. The first trial happened in 1990 at NIH. Then we had a period which there was kind of slow work through, in part because we didn't have perfect vectors. There was a death that occurred in an adenoviral vector gene therapy trial in 1999, which set the whole field back by probably 5 to 10 years, because there was kind of a reevaluation of what had gone on there. During that time, we had completion of the Human Genome Project in 2003, which ultimately has been an incredible boon for gene therapy, in part because not only understanding the human genome, but also because the technologies developed have resulted in the ability now to have a whole genome sequence for what is amounting to hundreds to a few $1,000s compared to the several $billion that the first genome cost. And then we had further advances both in the gene therapy vector field as well as in immunology that led to the first patients being treated with CAR T cells in 2010. And ultimately, we had the first approvals of gene therapies in the United States. They were approved outside the United States a little earlier, but except the first CAR T cells were approved here in the United States in 2017, along with the first virally vectored gene therapies. And since that time, there has been a fair amount of progress. But I think what's really impressive is that we have seen the transformative potential of gene therapy. We know that gene therapy is not always perfect, but when it works well, it is pretty amazing. And if you look at, for instance, Emily Whitehead, these patients that don't worry, no confidences are violated here. These are all patients who have put, and their parents who have put their names out there. So you can find this on the internet. Emily Whitehead was the first child treated on a trial for acute lymphoid leukemia that was relapse refractory with CAR T cells. A tremendous amount was learned from treating this one patient about of how these patients had to be managed, including the inflammatory reactions that can occur when you actually have a very effective therapy. Emily is now as a hematologist oncologist. I can tell you she's 10 years out. She's probably curative of her leukemia. Nico Nieves, child with spinal muscular atrophy, treated very early in life. One of the earliest treatments. You see a child there looking very happy, standing, walking. Anyone who knows the natural history of spinal muscular atrophy type 1 knows that's your diagnosis would have to be wrong if you saw a child standing there, because these children can't walk at this age, and they're usually not alive even by the time they're 2 to 3 years old. And then most recently, we had baby KJ, or KJ Muldoon, born with carbamyl phosphatase synthetase 1 deficiency, discovered very early on in life by whole genome sequencing. And then a bespoke therapeutic was developed over the course of a few months at University of Pennsylvania, was administered, actually was tweaked, administered a second time. And here you see baby KJ at 16 months now, doing quite well. Whereas again, normally children with this would either be severely disabled or dead. So really amazing transformative potential of this. And to it, in the United States, we now have 27 approved gene therapies. They essentially are across 3 major buckets. Cell-based gene therapies, many of them are chimeric antigen receptor T cells. Localized gene therapies, things given into the eye, or for instance, or into a given organ. Luxturna or Voretigene was the first of the locally administered gene therapies for RPR65 blindness, and again, a very amazing transformation. Children who couldn't see, the biggest problem there was they couldn't, you lose the ability to see in low light level situations. So children who couldn't go out after dark and play were suddenly being able to go out and play after dark. Maybe their parents didn't like that all that much, but that was another amazing transformation there. And then we have the systemic gene therapies, as I've already mentioned, one of them on onasemnogene for spinal muscular atrophy type 1, but a variety that have also followed. And one could say, wow, we have 27 approved therapies, but we've got a different problem. We have 10,000 plus rare diseases. So if we keep going at this rate, a former colleague of mine, PJ Brooks, calculated that it will take 600 to 800 years before we get to a therapy for all of these. So we obviously, now that's obviously, you could criticize how that calculation was made, but it's just an example of how we really do have to think about how we can do this faster. There have been some challenges that we've had with gene therapy. One of the issues has clearly been we've settled for systemic gene therapy a lot on adeno-associated viral gene therapy vectors. they have their own problem associated with them, which is when you give a big slug of adeno-associated virus intravenously, you get an inflammatory response. And when you give the really large amounts that we've had to give in some cases, that can be problematic. Getting more potent vectors, one of the things that is probably going to change things. And we're already seeing the introduction of vectors that are an order of magnitude more potent, allowing you to give fewer viral genomes. That's probably necessary if you want to deal with diseases like Duchenne's muscular dystrophy, where you've got to really address kilograms of muscle with your gene therapy. And you just have to have a potent product to be able to do that. Reducing the immune response to AAV, obviously important, because two problems with AAV. A, you don't like that initial immune response, which causes oftentimes liver inflammation. But you also have the issue that the immune response precludes you from giving multiple doses of AAV. So unfortunately, once you get antibodies to one AAV serotype, you're usually are refractory to or immune to all of the other serotypes that'll be given. So this is something that's being addressed with both novel immunosuppressive regimens as well as with the development of designer capsids that may escape immune surveillance. And probably the most exciting and promising of these technologies is the use of in vivo gene editing techniques or delivery of proteins via mRNA. Same general idea, the idea that you're using lipid nanoparticles, the same technology that was developed for the mRNA vaccines to deliver mRNA that will either express a CRISPR to genome edit or express an mRNA to express a protein. And there, some of the challenges have been on organ-specific delivery, but it's remarkable how in a few years we've now seen the development of lipid nanoparticles that target various organs, the most challenging of which still has been delivery to the brain, but even that is starting to be addressed as well. So here's where hopefully things will go. And I think we're seeing a lot of progress. I mean, I think even in the past few years, we've seen a lot of things that we thought would take longer to get over, such as targeting various tissues. We've seen progress there. But there's been a lot of challenges as well. Demonstrating safety and effectiveness in small populations is a giant challenge. And that's because we're dealing with small numbers of patients, sometimes a handful, sometimes dozens. And they're dispersed globally often. That's a challenge. We like to have natural history of a disease so that you can do a single-arm trial and compare it to the natural history. But oftentimes for rare genetic diseases, we don't have a well-documented natural history. Or even if there is a natural history documented, because of the nature of different mutations within a gene, you see a lot of variation, and it's hard to say that you have one phenotype to compare to. So this is a challenge. A very large issue is the complexity and cost of gene therapy manufacturing and administering it. The cost of manufacturing vector, depending on where it's made, can be anywhere, just to give you an idea, from $50,000 to $500,000 per dose. That doesn't include setup costs and all of the assays that go with it, which can often cost $5–10 million to set up. So this is a very expensive way to go about doing things, especially if you do it new each time, a new gene therapy starting from fresh each time between different diseases. The immunosuppressive regimens are such that these are not products that you can go to your local doc in the box and get. They have to be given at specialized hospitals like Rainbow, others. And one of the other things that we don't often think about a lot is that for the rare disease population, we have different regulatory environments in different countries. And despite the fact that we might have 5 patients with a given rare disease here in the United States, the European Union might have another 7, Japan might have 2 or 3. We each require a regulatory submission that's completely consistent with our own applicable laws. which means redoing all this work, which costs, again, millions of dollars, when at the end of the day, we all come to the same conclusion generally, that the therapy for these small populations is safe and can be used. So we need to think about how we address that. So I'm going to spend a couple more minutes now kind of trying to go through how we address some of these things. Because ultimately, we have to think how we can break through the barriers, limiting our ability to apply what we've learned about the genome to be able to effectively practice genetic medicine. And I'm going to back it up a bit now, and I hope I don't put everyone to sleep, but just to give people a little bit of an idea of what tools we have in the tools chest to get there, and also to try to make the case that what I'm going to talk about in terms of a more process-based approval pathway is not something totally novel. And if you look at the history of U.S. Food and Drug law, the Biologics Control Act of 1902, actually back in the day, laws were very concise. It's about 3 pages of an act, which actually has a tremendous amount in it. And it was put into place after 22 children died of the biologics at that time, which were diphtheria antitoxin and smallpox vaccine in Camden, New Jersey, and in St. Louis. And what it really did was it allowed an agency at that time, it wasn't the FDA, it actually was part of the Department of Treasury, to issue licenses for processes for making biologics. And ultimately, that was modified to our safety and effectiveness standards through the Food, Drug, and Cosmetic Act and the Kefauver-Harris amendments, which were the efficacy amendments that were put in place shortly after the thalidomide catastrophe. And then we had things that have really helped us with developing products for orphan diseases, the Orphan Drug Act, as well as the FDA Safety and Innovation Act in 2012. So just for those who may not be as familiar, Orphan Drug Act of 1983, back then it was hard to get companies to develop products for small populations. It seems crazy now, unbelievable, but back then nobody wanted to deal with small populations like population of patients with Gaucher's disease or Duchenne's muscular dystrophy, because at that time, all the money was in large volume products. Orphan Drug Act said, okay, population less than 200,000. It put in place incentives with a designation that gave tax incentives and a waiver of the submission fee to FDA for those products. And then if you got those products approved, it gave 7 years of exclusivity, which is a pretty big deal for a marketed product. That was built upon further with the FDA Safety and Innovation Act of 2012, breakthrough therapy designation, which wasn't specifically for rare disease products, but it's been used extensively for them, allows those products to take use of a number of different pathways, which I'll tell you about in a moment, that allows you to help move their development forward more rapidly. And then importantly, for small companies getting into this area, many of them spinning out of academic institutions, was the Rare Pediatric Disease Priority Review Voucher Program, where Congress set this up so that for products that are designated as being for a rare pediatric disease, once an approval comes for that product, the FDA issues a voucher. That voucher is a negotiable instrument, and it can be sold to someone else to convert a standard review, which takes 12 months, to a priority review, which takes 8 months. Now, you might say, why would anyone want to save 4 months? Well, if anyone looks at the sales of, I don't know, maybe weight loss drugs, 4 months of sales, it amounts to a lot of money. So $100 million for that 4 months turns out to be what these vouchers tend to go for. So the vouchers tend to be selling for $50 to $150 million for a small company developing, wanting to develop a gene therapy. That's a really good impetus for them to move that forward. Oftentimes they'll sell the priority review voucher, and at the same time, the company sold as well So it helps the ecosystem move forward. This actually expired for a while. It was a bit of a catastrophe. It was just renewed on February 3rd, much to everyone's gratitude. And so this will now be in place for another 5 years. The idea here is that FDA has a number of ways to expedite not just rare disease products, but all medical products that include things like priority review. That's what I was talking about, getting the review time down, accelerated approval, approving something based on a surrogate endpoint rather than a traditional endpoint of how someone feels, functions, or survives. And we have things like breakthrough designation, which I noted, and regenerative medicine advanced therapy designation, which is similar to breakthrough designation, but it's applicable to cell and gene therapies. All of these things have helped move rare disease therapies forward, but they haven't helped us make the quantum leap that we need to get to many different products. Now, how do we get there? And I think one of the ways we get there is we kind of step back and think about how we assess medical products. And this is actually from an FDA guidance that was published a number of years ago. When you think about medical products from a regulator's standpoint, obviously you want the benefits to outweigh the risks, and you want to be able to manage the uncertainty associated with the benefit and risk, because there's always uncertainty. When you think about it, for that weight loss drug, you want the benefits to greatly outweigh the risks, and you want there to be very little uncertainty. You don't want to find out that 1 in 50,000 or 1 in 100,000 people get liver failure from it, right? You want to have minimal uncertainty, maximal safety, and good efficacy. When you start to think, though, of smaller populations, that trade-off may change. First of all, the public health risk of treating 10 or 100 patients is not the same as when you're going to treat 1 million patients or 10 million or 200 million patients. This is a little bit of the risk calculus that I learned during the pandemic when we were, in fact, doing that. But you're also in a situation where we have to think about the benefits and risks and what patients and parents are willing to think about in terms of the risk of doing nothing, which oftentimes can be death or severe disability, versus the risk associated with a therapeutic that may not be 100% effective or that may have some side effects. Now, interestingly, when you come at this from the hematology oncology perspective, we do this in cancer all the time. But for rare disease, making that leap has been a little bit more challenging. And although we can't throw out the safety and the efficacy to the point that we are doing risky therapies, I think we have to reassess or retune our risk-benefit calculations. And we also have to take advantage of what rare diseases that have a genetic basis have going for them. It's true they may affect small populations, which is in some ways helpful from a public health perspective because we're not developing these things for large populations. We're able to find them now increasingly with whole genome sequencing. We do know that if you fail to intervene, you may miss a window in which you can prevent irreversible deterioration or death. We don't have therapeutic options available for most of these, but we do have technologies available like gene therapy that can actually address many of these mutations. So what are we going to do here? Especially because you can assume if something is missing and you're able to replace it most of the time, that you might reasonably be expected to provide some benefit. Not always the case. Again, I'm kind of generalizing a bit here, but I think this is a reasonable assumption to a first approximation. And therefore, the benefit risk considerations are not infrequently favorable for genetic therapies. However, we're not able to get there because genetic therapies are generally not readily accessible because of their cost and complexity. So can we take advantage of advances represented by genetic therapies because of their unique characteristics that you can exploit in the drug development process? Can you look at just understand you've got a fundamental mechanism of action that's understood. This is not like a small molecule that's doing something magical. This is a product that's interacting with the genome on specific nucleic acids, or that's providing specific nucleic acids to produce a product. We can actually find something that's kind of nice about these that's fundamentally different from many small molecules. Small molecules, generally, when you switch one atom on them, can change their properties massively. If you look, for instance, the difference between something that's highly teratogenic, like thalidomide, and something that's not teratogenic or much less teratogenic, like lenalidomide, is one atom. Whereas for gene therapies, we can take a backbone vector and put in a lot of different inserts. And by and large, the manufacturing process and the production process, and for that matter, the administration process, is going to be very, very similar. And so we have not taken advantage of that fully to date. And that's something that we could potentially do. And when you think about it too, with AAV therapies, the toxicities are pretty similar, just like for CAR T cells. Toxicities, pretty similar. We have problems with cytokine release syndrome and ICANS, or neurologic events. So this, I think, starting to think about what we can take advantage of is important, because you can start to think about other ways of doing biologic development that takes advantage of a process-based approval approach. And this process-based development and approval approach does mean you have to balance benefits and risks, but you're generally doing this where there are no good alternatives. You're not going to be doing this for acne, okay? This is not, I would not advocate this pathway for new acne medications. On the other hand, for a life-threatening rare metabolic disorder that will kill a child by the age of 3 if they don't have something to fix that, this is not such a crazy idea if we could treat more of these. And that's the idea of having a pathway where you would have reasonably compelling non-clinical evidence, safety, and efficacy data for a group of products. So you'd basically show that your manufacturing process, your delivery process, your backbone vector, that it works for several products in a class. They would all be of the same thing, AAV8 vectors targeting the liver to produce a protein. And what you would then do is get an approval. We would approve a product for that general class of treatment rather than for the individual diseases. And what you then do is have the companies be able to, or the academic institution be able to switch out the inserts within reason. Obviously, you'd have specifications for what you could do, and not have to go back to FDA each time. They basically would go back to kind of what was going on at the turn of the 19th to 20th century, where it was more of a process base. And obviously, you'd have to keep track of safety and effectiveness of those products. No one's saying that you would just send it out into the wilderness, but that would allow one to potentially move and address more of these therapies with, I think, a reasonable benefit risk calculus. And obviously, you'd have to have appropriate oversight, et cetera. But this is a way of potentially thinking about getting there that builds upon the current plausible mechanism guidance, that a draft guidance that was just actually released by FDA earlier this week. It's been in the making for several years, and hopefully would allow us to move forward. Obviously, a lot of tweaking here is necessary, but we have to think about some way that we can move forward more quickly, because right now, if it's going to cost hundreds of millions of dollars each time we want to develop a product for 10 people or 20 people, it's just not going to happen. It's just not going to happen. So this is a way of getting there with, I think, a reasonably balanced benefit of risk. Another thing that we might be able to do here is try to reduce the cost of gene therapy by this. It goes without saying that if you don't have to do all the toxicology work and all the manufacturing process development each time, this is a way that you potentially could reduce costs. You also can reduce the administrative burden here, because it turns out it costs a lot to submit things. You have to put a lot of people to work submitting regulatory filings to FDA. And so you might be able to reduce a lot of those costs, and you might also be able to take advantage of some other things as well. AI can actually help us a lot in this in terms of both manufacturing and also in terms of some of the regulatory burden. And advanced manufacturing technologies are also something that's important because we need to just try to reduce complexity of process development and improve efficiency. It may even be possible, and this is a prototype that was developed for instance, for the lipid nanoparticle-based mRNA constructs, to make these almost in academic institutions at small scale in an automated manner, again, greatly reducing the cost. And you could, if you weren't having to go back to go through the entire process as a new product each time, but rather have a process that was approved and you were keeping to that process, getting safety data, efficacy data, submitting it over time. This may allow us to address more diseases. And then finally, just to come back full circle, this issue of having different global regulatory environments has really been a particularly challenging issue with rare diseases. I mean, it's a little bit crazy to have a product approved in the United States for 10 patients. Europe has some products that are approved for 10 patients or 20 patients there. And to not be able to have our populations benefit from these because of the different regulatory environments, when in the end, most of the time, the very large part of the time, when they're submitted to the other regulatory agencies, they're approved. And so this just becomes a barrier. And there might be a reason to have these kinds of barriers, market barriers to entry for large products that cost healthcare systems a lot of money. But these basically actually cost healthcare systems in each country money because they don't exist. And so the cost of supportive care, taking care of a child who's severely disabled, probably costs more than by far than having these products available. So one idea is if we can get to some global regulatory harmonization with essentially almost mutual recognition of approvals, this could help get these products to more people. The WHO, World Health Organization, has been working on this. We started working with them probably 7 or 8 years ago on trying to get towards the convergence necessary for this. It's been slowed down a bit, but hopefully this will take root again because it could make a big difference in the rare disease space, could improve lives and make a difference here. So if you put it all together, it's still kind of early on in the era of genetic medicine. I mean, I have to say, we are a little bit, it's great to be here with babies and children because we are still kind of babies and children in the gene therapy world. We're not adolescents yet. I think I would not say we're in our adolescence, but it's still exciting. And we are starting to see a stream of products. I think if we can kind of get our heads around using a pathway that's different from the one we use for large volume products for these rare disease products, it's not fundamentally different. We still are thinking about safety, efficacy, benefit, risk, uncertainty, but it does have some different properties. We can hopefully get to treating more patients more efficiently and making a difference in people's lives. And I think that's warranted by the fact that we have a different understanding of genetic medicines than we do about many other types of medicines. So all of that coupled together with advances in technology can hopefully help us make a difference for patients. And I will stop there. Thanks for your attention, and I look forward to taking some questions.
That was great. Thank you. And I needed that review. I feel so much smarter after that half hour than I did walking in the room. I will also say that not yet being in adolescence is probably not a terrible thing as a dad and a pediatrician. I think that elementary school age kids are the best people on the planet. Blossoming personality, intellect, wit, and yet they still have that sense of wonder and magic, and they want to please everybody. So that may be a sweet
That maybe, yeah, no, maybe we could send them away from about, I know I have two 20-somethings. If they go away between about 11 and 24, that's probably okay, right? No, sorry.
Two questions. It was very good, and I agree with Mike. So first question is part of the global approval process complicated by proprietary interests of the companies? That's the
Yeah, no, that's a really good, it's a really good question. So you'd have to basically have, are there global competitors that would create an issue there? In general, in this space, there's not. That's because nobody wants to actually, it's just too expensive, so that you don't have that type of competition. And in fact, the few, what I would call catastrophes that have occurred in this area of gene therapy have when you've had 2 or 3 companies working in the same very rare space where they essentially annihilate each other. So the good news is, in general, it's not a problem, and I think it's one of these things that's worked its way out. But the problem has been less about the corporate issues and more about different regulators wanting each of their check boxes checked, because there, over the course of time, subtly different or not so subtly different requirements for gene therapies have developed in different countries. There is actually a great precedent for how we could get there, which is that in the vaccine world, the World Health Organization has pre-qualified vaccines. Basically, if a stringent regulatory authority like the European Medicines Agency, the FDA approves a product, you can submit to the WHO. They then list the product as one that has been vetted. They also do some additional vetting. And then a lot of countries, for instance, Africa will go and look at that, and they don't have to go through and have their regulators approve it. They basically take the approval from WHO. So it might be something like that for gene therapy could happen. I worry less about, I worry less about anyone caring about the competition for these and more about just getting through some of the regulatory barriers.
The second question is gene therapy and infrequent disease synonymous? Is every rare disease always single gene related?
No, obviously that's an incredibly challenging problem because there are many. That's why I try to give some caveats here. There are obviously some rare diseases that are multi-gene or there are whole pieces of genome missing. And people are working to try to address that. Some of them are trying to address that with very nifty methodologies of using CRISPR and long genetic elements in mRNA constructs to be able to replace whole sections of genome. That's probably a decade or more off. Although every time I say, my problem is that's like the kiss of death for me, because every time I say something like that, next week you'll read about it in Science. So, but you're absolutely right that there are a variety of genetic diseases that are much more complex. That's why I say we're still in our childhood, because we still have a lot of more complicated diseases, probably a bunch of diseases that don't involve coding sequences, that probably are diseases based on intronic issues that lead to pathology that we just don't understand yet. But the good news is that we do know, I've kind of done an inventory of this, we do at least have hundreds and hundreds of single gene disorders to work on. So while we're discovering the other things, we can keep ourselves busy.
Part of the big.
There's a question online. They want to know, have you proposed to the shared pathway to the FDA? And if so, has there been any response?
So I have not, but there are others who we were working on this before I left FDA, and there are others who are approaching FDA with this type of a pathway.
You mentioned the cost of production of the gene therapy vectors. Can you, since AAV production is entirely scalable and with advanced manufacturing, is the cost the regulatory issues, or is it actually the manufacturing of it itself?
So it's actually, it's a great question because it's a combination of both. The manufacturing, because you have to make it in, you're using a producer cell line and you have to tweak that. It's not an inexpensive process. But the problem that ends up happening now that adds a lot of cost is each time one of these are developed, all these assays have to be redeveloped. And it costs somewhere between $5 and $10 million each time you go to develop a whole set of qualified assays to document that the production of that particular AAV is acceptable. And that's repeated over and over again. By the way, it's a business model that contract manufacturers use. And we can go in private. I'm happy to have a discussion with you about my personal feelings about that business model. But that is what's adding. So you're right. It's the actual production cost. It's not the worst piece of it alone. But it's not. It isn't cheap to make AAV vectors, but it's not super expensive. What really adds on to the cost significantly are all of the accoutrement in terms of the characterization that has to happen.
The process-based approval approach is very compelling, and I'm wondering if there are diseases that inherently would be resistant to that process. There's something about the disease itself that makes it not feasible.
Yeah, well, I think you're exactly right. That's why, again, I caveat that there are going to be there are going to be exceptions that are not going to be appropriate for this. But of what might not be appropriate, diseases that might require a variety of different. There are some diseases that might have different point mutations and even hemophilia is a great example. You have point mutations, you have an inversion mutation. You might not be able to get the same kind of. You're probably going to have to use different approaches there. So this kind of process, one process isn't going to take care of all of hemophilia, unless that process adds a new gene, to a different place. But if you were just talking about fixing the hemophilia gene with a genome editing approach, probably one process wouldn't fix it. I probably, if I have a little more time, can think of better examples. But it's okay. I mean, I guess as somebody who does a lot of carpentry, I have a lot of different tools in my toolbox, and I like that. Okay, there are some that I take out very rarely, but when you need, a Japanese saw, it's nice to have one because you really need it. So I think that's a little bit what I would think, that I don't think this is going to fix everything, but if it took care of some of the more common things, point mutations or nonsense mutations, it would potentially help speed up the course of development for that many more patients. Because when you're dealing with 10,000 and we have 27 approved therapies, we got a lot of progress we can make. And even if we can't get all the way there, getting a quarter of the way or halfway there would be an amazing thing.
Moshe Perot, Pete's Palm. Thanks for the talk. I'm wondering, you showed 3 examples of remarkable cases where the impact of gene therapy is evident within months to years. I'm wondering what your sort of global thoughts are on using, as you mentioned, surrogates of outcomes where you use biomarkers, because you just don't have the time in terms of these disease progressions. You're talking about natural histories that might manifest in decades as opposed to months and years.
Yeah, no, I mean, I think we really have to think about, so a different talk today could have been one about surrogate markers, where for any number of diseases where you end up seeing significant decline over time. For instance, lysosomal storage diseases, particularly the ones with CNS manifestations, they're looking at biomarkers as critical, because if you were to try to wait and do a 3-year study, it's 3, 4, 5, it takes that long to see the deterioration and to see that you've altered it. Now, some of those studies have been done now, And they're a little bit painful because it's very hard. You can't, putting a child in a control arm there is not, it does not feel ethical. So looking at biomarkers turns out to be really important. Having good biomarkers is a whole nother issue of getting the best ones. For instance, it took a long time during my days at FDA to get heparin sulfate to be accepted as a biomarker. And even now we're revisiting it, and we're seeing it revisited at the agency. So biomarkers, very important. And it's particularly important because many of the diseases that will be addressed by gene therapy are slowly progressive. And the other key, though, when they're slowly progressive is that once the function is gone, you can't get it back. So trying to be able to intervene early is important.
We have some patients that are even admitted to our hospital currently who are now adolescents, young adults with multi-system organ disease. And I think in kind of more recent years, we found that they do have some genetic mutations. And I'm just thinking forward, like, what would be your recommendation in the coming years about how we approach these kids when we see them when they're young? Like, who do we send for genetic testing to try to get ahead of some of this long-term disease?
Yeah, so I don't know that I have the perfect answer for that, but I can tell you that one of the really interesting figures that somebody I know works in this area in the pediatric NICU, when they have kids that are immediately have issues after birth and need intensive care, and they're not able to immediately come to a diagnosis. If you do whole genome sequencing, you actually get a hit rate about like 8 to 10% of diagnoses of rare genetic syndromes. Whole genome sequencing is very powerful. I don't know how you do it. It's the issue. Now I guess I'm getting the oncologist in me is coming out. If you do it in an adolescent and there's not a lot you can do you even do the test? Because if you can't intervene, is it worth doing a screening test? But I think it's probably a message to us that in this age when a whole genome sequence is less than an MRI read at a radiologist. I'm talking about Washington. I don't know what Cleveland cost is, but I know if you go in the Harvard system or in to Georgetown Hospital, an MRI costs more than an MRI with the reading of the brain costs more than a whole genome sequence. That probably the idea here would be that if you even think that there could be a genetic syndrome early on as the child is developing, the earlier the better to do whole genome sequence. There are countries that have started to consider whole genome sequencing of every baby born because of the cost issues and what you can get as a hit rate. We don't particularly like that here in the United States because of the issues of Big Brother. But that's another story for another day.
There's another question online. Do you have any sample verbiage and a list of those in charge to whom we can advocate for the more streamlined approach to creating treatments for our diseases?
I couldn't hear the first part of the question. Sample verbiage. There is verbiage in development by a former acting commissioner and former director of CDER that I'm very well aware of, Janet Woodcock. So there are people working on that. I think it's the Haystack group that's working on this. I know I've interfaced with them. Great. Thank you so much.
Coolest audiences and one of the most interactive Q&A sessions we've had in a while. So thank you to you guys for showing up and thank you for leading such an engaging discussion. We do have a meet and greet in the community room. So if you'd like to continue this conversation and grab some food, please join us just a few steps down the hall. Have a great day.
Thanks so much.